So I want to thank the organizers. It's always nice to come to Paris, and this is a great meetings. You know, we're starting to see a lot of meetings on microbe interactions and how they impact interactions with the host. And so, but today what we're going to do in my talk before lunch, I know I'm sort of between you and lunch, we're going to step back a little bit. We've been hearing a lot of really nice mechanism over the last few talks, and so what we're going to do now is sort of step back and sort of start thinking a little bit about ecology and maybe a couple of other things. And hopefully by the end, what I'm going to probably, at least hopefully try to do for you, is provide you with sort of a conceptual framework for the way my lab thinks about microbe interactions, and maybe an experimental system, and maybe try to get at some questions that I don't know that have really been addressed in microbiology yet. So what we're going to talk about are polymicrobial infections, and obviously I'm not the first person to think about polymicrobial infections. There's actually a lot of work in France and Germany in the mid-1800s on this subject. It wasn't really concerned with the fact that they would look at infections and see there were multiple microbes that actually were inhabiting these infections. What they were actually doing was actually pursuing this idea that you could actually use microbes to cure infections. And there was a lot of this actually going on in particularly in the 1860s, where folks were actually trying to isolate microbes and actually asking the question, could they actually outcompete other microbes in the lab, particularly Bacillus anthracis. And there was a really cool paper that actually came in 1877 and by Pasteur and his colleagues. And what they actually did was a really interesting experiment where they were studying Bacillus anthracis. And they had actually found a microbe from soil that they found in the lab could actually outcompete Bacillus anthracis in the lab. And then what they did is they went into these animal models, and they were using guinea pigs and rabbits, and they actually showed, now they didn't have to do as many animals as we do today to actually publish a paper, but they were actually able to actually show that you could put anthrax into these models of infection. Of course, the animals would get sick, but you could also put in spores of Bacillus anthracis and wait 1 hour and put in this other bacterium that they knew in the lab would outcompete anthrax. And they were able to actually cure about 60% of the animals with this. And there was a lot of work after this. A lot of people seem to not believe this because this was sort of, I guess, a paper that what you see immediately after this in the late 1870s and early 1880s, what you see are large numbers of people actually repeating this experiment. And one of the bugs that they ended up using for this experiment is one I'm going to actually talk to you a little bit about today, and it's called Pseudomonas aeruginosa. So what you're actually looking at here is this is Bacillus anthracis on a plate. This is the Stern strain that I'll let my undergrads work with. But on this plate, and then what you can see is you can spot some Pseudomonas, and you see this big zone of clearing, and we can find mutants that don't do this. And so this was really sort of what people were thinking about sort of at this time. But this sort of spurred a lot of interest, and primarily from physicians. And what you started seeing are a lot of people publishing papers on clinical infections. And these are just a couple that I want to show you. But in the late 1800s, people started to realize one thing, and it became very pervasive in the literature. And that was that if you had an infection, and you had microbes in that infection that would grow with oxygen, and those that would grow with low oxygen, that the infections seemed to be worse. That you seem to have a more intense infection. They didn't quantify bugs, but they were just quantifying clinical condition. And some of them would actually some of the physicians would actually go so far as if you could actually do that, what you would do was actually sequester those individuals away from others because you knew they needed to be looked after more. And so this was actually a really cool paper, actually. If you, I obviously don't speak French or German, I speak Texan, and I think that if you get these translated, what you can actually see is what these folks were doing were pulling appendicitis out and smelling it, and if it smelled putrid, they would actually say, oh, we need to put these people sort of separately and monitor them more. These are probably volatile fatty acids, right, that the anaerobes are making. So they didn't need a mass spec, right, to actually figure out what to do with patients at the time. And they were just looking for putrid smells. What you had was sort of a lull sort of in the early 1900s, or at least what I can find in the literature. You had this real lull in the study of multi-species infections. And it wasn't until about the 1950s and 60s, and it wasn't the gut that was doing this, it was actually the oral cavity. So it was people working in the mouth. We can grow most of the bugs that actually reside in the human oral cavity. And what people actually started doing was just taking bacteria from the mouth and randomly mixing them together and putting them in some infection model. Most of the time, this is an abscess model. And what they found was something that's really interesting. And it wasn't that you could actually just randomly mix bacteria together and the infection would be worse. But they found that there was a high degree of specificity. And it took like very specific combinations of bacteria to actually cause these sort of worse diseases that we are thinking of now in today's world as polymicrobial infections. And actually, in fact, what they would see most of the time that they would actually put bacteria together in these infection models is the infection was cleared much faster than it was for the mono-infections. And probably from some of the ideas that Kevin Foster was telling you about, and these idea that these bugs are actually competing in these infections. But I want you to remember this concept of specificity. So this is the sort of question we're going to talk about. Why are these infections more severe? The ones that are, why are they? Focusing on microbe. We do some host stuff in my lab, but today we're going to focus on this. So I like to go through this exercise because I think you're starting to see a lot of this actually in the literature. And well, how do you choose the bacteria that you're going to study in the lab, right? So what do you do most of the time? You get some clinical sample. Maybe this is sputum. This is sputum from a cystic fibrosis patient. And what do? You sequence it, right? And then you say, oh, well, these bacteria are there and these bacteria are there. Well, they must be interacting in disease. So I would actually argue that it's a little bit more complicated than that, and we should probably be thinking about things a little differently and maybe a little bit more thoroughly. So you might imagine, so a CF sample can have 10 to the 9th bacteria. We think this is a lot of bugs, right? But bacteria are small. I think we sort of forget this sometimes, at least from the clinical standpoint. E. coli, the volume of an E. coli cell is about 10 to the minus 15th liters. E. coli is big compared to a lot of the bacteria that I work on. So if you have 10 to the 9th of those, you have about a microliter volume in 1 mil. Okay? That doesn't sound like that many, right? And so when we start thinking about this, what we have to think about more than I can just get a clinical sample and sequence it. But you also have to think about, are these organisms actually spatially localized so that they would be interacting in these infections? The other thing that I thought about a lot for the first maybe 5 or 6 years of my lab, we were doing everything in vitro. So we did a lot of this. This is a beautiful picture of Pseudomonas aeruginosa, this beautiful green color. And then these, of course, are flow cell biofilms from my lab. And we really started thinking about how relevant are these systems. And what we found, at least in our hands, is a lot of the interactions that we would actually find that you could recapitulate in these systems, we could never get to recapitulate in more natural systems. And we tried a lot of different ones. And so why was this? Well, probably the growth environment's very different. Maybe it was the fact that most of the time we were just studying Pseudomonas on its own and asking how it interacts. And so we've now gone to 2 systems I'm going to tell you a little bit today about, and these are using mouse models. And we probably all know there's a lot of people doing mouse work in here. Not all mouse models are the same. Some models are a little bit better than others. Some are a little bit more relevant than others. And we've searched pretty hard. We think these are pretty good. I can tell you why later, but comparing sort of transcriptomics in a human to transcriptomics in the mouse, I think a lot of times one can actually begin to ask questions about whether the bacteria are behaving the same in both. And so what I'm going to mainly talk to you about today is this binary system of Pseudomonas aeruginosa and Staph aureus. You've probably seen this a lot. These bacteria co-occur in many, many infections. About 80% of wound infections have both of these organisms. Wound infections, which is what we're going to talk about today, are about 12% of the U.S. healthcare budget right now. Chronic wounds, long-term wounds that last months and years. And these bugs are the primary conspirators in those diseases. And we have mouse models for these infections. We have acute wound models and more chronic. And we can this is the one we'll sort of talk about more. I also work on oral microbes, and so I know a lot of people, and I think maybe Karina said that, you know, the gut is like so really important, and I agree with that, but I think that also the oral cavity is also very important. And studying the oral cavity probably has a lot of advantages that the gut doesn't have when you talk about human studies. One can actually get samples very easy. One can look at spatial localization. One can actually do lots of manipulative studies in those systems. And I think it's even more relevant now because what you're starting to see, there's going to be some studies coming out that show that most of you probably have bacteria in your bloodstream right now, probably if you brushed your teeth this morning. So almost every time you brush your teeth, you have a small number of bacteria from the oral cavity that actually can be cultured out of your blood for about 2 to 3 hours after you brush your teeth. And a lot of people are now beginning to think that these bacteria are localizing to certain areas and are important causes of things like endocarditis. So the bugs I'll tell you about today, I'm only going to give you one slide. We use an abscess model of infection, and the bugs are this bug here called Aggregatobacter actinomycetum commodans, so that will be AA from now on. And then this is a commensal that all of you have in your mouth. It's called Streptococci. And we use an abscess model, which these bugs collectively together, you can find them in abscesses in humans. So we've been doing a lot of this for a while, maybe the last 5 to 6 years. We've been actually looking at interactions, and we found a lot of things that are important. And sometimes we started in vitro and went all the way to animal models. Sometimes we just went to the animal. And what you can see is with these systems, one can see interactions. Most of these interactions, and I believe probably most interactions between microbes, are probably mediated by metabolites. We see a lot of this in these systems, but you also see things like outer membrane vesicles and peptidoglycan being really important ways in which microbes can actually sense another and have very specific responses to these. So I think all of this work actually came from an idea that we had that, well, maybe that probably what I believe that what we thought and we hypothesized that most microbes in the environment are not necessarily looking for signals. Signaling is actually fairly hard to evolve. I think what you're doing is looking at biochemistry around you, and the way that you assess the biochemistry of the microbes around you is often with metabolites. And this is sort of how this started. The problem with this sort of approach is it's been pretty slow. It's very difficult to actually figure out signals, find receptors, and these sort of things. So we've moved to more large-scale studies, more systems-based approaches, and that's what we'll talk about a little bit today. So the one I'm going to talk about today is something all of you know about. It's used a lot. We call it transposon sequencing, or TNSeq. It's a very simple conceptually. I'm going to go through it with you just to make sure, because we're going to use it a few times. So basically what you do is you take your bacterium of interest, this can be Pseudomonas or Staph, and you make a transposon library where these colors actually represent a strain that has a transposon in a very specific location. We usually try to saturate the genome to about every 20, so you have an insertion about every 20 base pairs. So in Pseudomonas, that's a couple of 100,000 mutants. In Staph, it's a few less. And then what you can actually do is take this population, assess abundance of every mutant, do some sort of lightning bolt symbol, which could be anything from an antibiotic, which we've done, to animals, and then you assess abundance again. And the idea being, and you do this with a really simple sequencing-based approach, it's pretty simple, and the idea being very simply that what you're doing is looking for mutants that are abundant here and change in abundance here. And we do this with the number of reads that you get. And so if you get the same number, if you get a sort of a relative abundance of a mutant that's the same here, maybe this green one in here, then you know there's no, it's not a fit, that insertion is not a fitness determinant, but maybe if for this white one and the gray one where they disappear, it's a fitness determinant. Again, you guys probably know this, but it's a way to assess fitness. So what we've been doing is actually going back to these systems that we worked on, these wound systems, and asking very simple questions, and doing this fitness profiling with the question of trying to identify genes that are important for fitness in mouse infections. And so we've published this monoculture, and I think it's really interesting. What you can do is you can actually go in and you can actually, by looking at biosynthetic pathways, you can actually say, well, we know in the wound that these nutrients are there and these nutrients are not there, just based on the fitness of particular biosynthetic mutants. So you can learn a lot, I think, from this. And we've done the same with Staph aureus. And what I'm going to talk to you about today is Staph aureus. And I'm going to tell you about what genes are essential in vivo. And so when we talk about essentiality, you can look at this paper from a postdoc in my lab, Keith Turner, but when we're talking about essential genes, we're talking about genes that are only essential in vivo. So these bugs will, you could insertionally inactivate this gene, that bacterium will grow on a Petri plate, but when you put it in vivo. And Keith came up with a Monte Carlo method to actually identify essential genes. And there's a lot of ways to do this. So, but just think about when I say essential genes today, it's genes that are really important for growth in a mouse. So we've done this with Staph aureus, and I wanted to show you some data. None of this is published, but I think it's kind of interesting, and I think it's kind of a way at least my lab is going to be going in the next few years, and that is there's actually been 2 Staph TNSeq papers that are published. Mike Gilmore actually provided the TNSeq library, and they did this in abscesses, which generally in humans are monomicrobial. Sometimes you'll see multi-species infections. Osteomyelitis, again, a lot of times they're monospecies infections in humans, but can be microbial, and then these chronic wounds, which are also polymicrobial. And so what Carolyn Iverson in my lab did is she took this data, and then she generated data in the wound model, and she just simply asked the question, how does infection site impact essentiality for Staph aureus? And what you see is something which I think we probably would have all predicted that you would have gotten. There's a set of genes that in all models are essential. So no matter where if you're on the bone, you're in an abscess, or you're in a wound, Staph is less fit if you inactivate these genes. But then there are specific genes for each one, and then some overlap between them. This is kind of, I think, what one might expect when one was doing these experiments. But really the questions we were wanting to ask, well, what happens as you build or increase diversity in the community? So what happens when you actually take Staph aureus and co-cultured in the wound with Pseudomonas? What actually happens to these numbers, and how do these numbers actually change, and how does the essential genome change? And so this is the question we're asking: how does the presence of this organism impact the fitness determinants in this organism? So this is exactly what you saw a second ago. This is a Venn diagram. Again, all we've done now is add on this red here. This is the fitness determinants for Staph when it's actually in co-infection with Pseudomonas in a wound. And there's two things that I want you to look at, which I think were actually a little surprising but very cool. The first thing that you see is that there are 461 genes that are essential, by our call, in Staph, but only when it's in co-culture with Pseudomonas. So this is about 20% of the Staph genome becomes essential when you actually have one organism that is now in. What are these genes? A lot of these genes are involved in metabolism, and a lot of them are involved in our stress responses. Pseudomonas induces a really strong competitive response in Staph. They compete for nutrients, and it's a much more stressful environment in those systems. So the other thing that's circled here is this gene. So what these two genes here are, it's 157 and 46, and what these are these 203 genes, the genes that are important in every monoculture, okay? So we go in here and you can actually look. Well, what you actually see is something that I think is actually pretty interesting, and that is 157 of those genes are still essential. So that means that Staph, whether it's in any of the three models or with Pseudomonas, it needs these genes to be fit in vivo. But what you see is a large number of these genes, which were essential, are no longer essential. Pseudomonas is actually alleviating essentiality. So genes that are essential in monoculture are no longer essential. And what you actually look at, you say, well, what are these genes? In fact, a lot of these genes are hypotheticals, but a lot of these are therapeutic targets. There's two real particular therapeutic targets that there's been a lot of work on that are no longer essential when one actually puts Pseudomonas into these infections. And so we've started doing this with lots of bacteria, and we call these sort of community-dependent essential genes. This is not published yet. Hopefully it will be soon. But I think if you look at this, it sort of makes you start thinking a little bit about something I mentioned earlier and some of the ideas that Kevin might have put forth at the beginning. And so Kevin actually put forth, oh, sorry, that's not Kevin. And that's not Kevin. That was when Kevin was in Texas, I think. Okay, so there's our Oxford professor. So Kevin actually, and Dan Cornforth, who's now in my lab, actually talked a little bit about this idea of competition sensing. And we see this in these competitive genes and these stress genes in Staph aureus. And we also see this in other systems. So the other thing I wanted to sort of bring up today, and something that I wanted to sort of give you an idea about what we're kind of interested in my lab, is something that I started thinking about in 2010 when I was at an ISME meeting in Australia. And there's a microbial ecology meeting, which is really great, and it's really fun to talk to environmental microbiologists because they think about things very different, I think, from the pathogenesis folks. And this word about biogeography kept coming up, and to be honest, I kind of knew what that was, but didn't really, so I went back to my hotel room, and you just Google it, right? And you ask, what is biogeography? And these are kind of the things that come up, and it's where birds are or where fossils are. And I started thinking, well, what about microbial biogeography? So you search that, and you get things that sort of look like this now. And you see this is probably a lot of fun. You're probably sampling here and here and here and here, and you're saying what communities are in these places. We've now even started doing this on our face, right, and on different parts of our body and our skin, and saying what organisms are in certain locations. But the idea is to think about what organisms are in space and time. Now, of course, these are really macroscopic examinations, and I was really interested more inside wounds and actually asking the question, do these organisms that I'm interested in, do they spatially organize? And this is actually a confocal micrograph through an infection. We've done some of this with human infections. This is a mouse infection. What you see is Pseudomonas in red, staph in green. There's a couple of things that you can probably notice. First off, they seem to form more clonal populations. You see these little clusters, and they look to be sort of segregated. So there's a couple of sets of data. If you sort of look through these types of infections, we see the same thing for our Aggregatobacter infections. Is what one begins to see is that in these soft tissue infections, you don't see mushroom biofilms, you don't see individual cells. Instead, what you see are aggregates, groups of high density of cells, and they're usually about a picoliter in volume, which is about 2,000 cells for staph, and about 1,000 cells here. So we have two computational platforms, and one can actually ask the question, so these aggregates, they're certainly growing in aggregates, but are these aggregates just randomly spaced, or is there spatial structure? And what we can actually see is there's actually very strong spatial structure, and in fact, almost all of the density that you'll see for the microbes is actually in a very precise spacing, where aggregates of staph and aggregates of Pseudomonas are spaced at an interval about 10 to 25 microns, depending on their size. So there's a very strong tendency for these organisms to interact and to be spatially organized. So we started thinking about this. This is a pretty tough question, though, right? So we can see this structure, but the question is this structure important? We know they seem to be organized. I can tell you they are. I can even tell you in what ways they are structured. These communities are structured, but the question is it important? This is a very difficult question to answer. It was actually initially answered somewhat by Apollo Stacey, who was in my lab. And Apollo wasn't working on Pseudomonas and staph, he was working on this other infection. But what Apollo was actually able to show is that these organisms also space. It's different from Pseudomonas and staph, but they space. And he was actually figured out why. And so what he learned was actually strep actually makes AA's favorite thing to eat, which is lactic acid. So you would think AA would benefit from being close, but strep also makes an antimicrobial that kills AA. And Apollo was able to actually go in and show that this spacing is actually highly dependent on peroxide. And AA has a mechanism to sense peroxide concentration and then disperse when it gets too high and reposition itself at specific intervals. And he was also able to make mutants to show that if you disrupt this system, and so make AA grow very close to strep instead of further away, what you see is there's essentially no infection now. So it's cleared within a day. So Apollo was able to sort of test this using genetics, but it's probably not the best way to do it, because a lot of times when you look at these interactions, the genes that you would actually have to disrupt so that you could disrupt these interactions are pleiotropic. They have lots of effects. So we wanted to have another way to do this. And so what we really wanted to do was to be able to spatially structure these populations and study them. And so I started thinking, well, how do you do this? I want to spatially structure aggregates of 1,000 cells specifically from one another. And if you actually go into the plant literature and actually ask the question, well, how do they study biogeography and spatial structure? They usually write an NSF grant. They usually pick a really nice island somewhere, apparently, because that's all the pictures I see. And then you go in and you remove plants, or you plant plants, and you ask how it impacts fitness of those around it, right? And so this is what we wanted to do. So we paired up with Jason Shearer. Jason is in the analytical chemistry department at UT. And this is Jody Connell, who's a postdoc in my lab, and Jody has a job. And we wanted to try to do this for microbes. So how do you do this with microbes? And the way that we've been doing this was by building little microbe houses. And so what you're actually looking at is something we call a bacterial lobster trap. And so this is actually a glass slide here. And what you're seeing, these are obviously walls. These walls are made of protein. So these are proteins that we have covalently linked by a process called multiphoton lithography, which I'll show you in a second. But essentially, all you need to think about today is we can use proteins like bricks to build very small things that can house bacteria. Okay, and this, what you see is this is an SEM, and this is Pseudomonas that was actually in this. We caught one Pseudomonas, allowed it to grow up. This trap is about 2 picoliters in volume, so this would hold about 2,000 cells. And you can kind of see the false colored green. So This is the process called multiphoton lithography. This is sped up 2 times, but this is about real time is kind of what you're seeing. So what you're seeing is the fluorescence that's actually emitted from a laser that's being scanned across the surface. And what you're doing is you're scanning across the surface, but what you have in front of the laser is a mask that's telling the laser where to go. The laser actually pulses in a 3D pixel called a voxel. It pulses, and then when it pulses in that voxel, it can cause cross-linking of proteins, and then you basically raster scan up and build what you want. This is a 5 picoliter trap. This is a pillar to hold the roof up, and this is a door to let the bacteria in. And so the bacteria that we've used to work on this, actually, they're like these very much. Well, they don't tell me that, but I mean, they grow. And so we catch like a single bacterium. This is Pseudomonas. You can close the door, and you can watch them swim around, and you can do certain kinds of studies. The problem with this technology when we first developed it was the fact that we had doors. And doors are not useful because doors have to be closed. And oftentimes what we had was we needed to let bacteria swim in through this door so that we could capture them. And so it's not really useful for non-motile bacteria. And so what we've actually done now is actually change this system now. So it's called and so instead of actually fabricating like we used to, we're fabricating in a 3D printing technique. And so we call this micro 3D printing. And so here's how this works. And this is really, I think, the advancement that Jody made that's going to allow us to do some cool experiments. So what you actually have here is some Pseudomonas, and then what you have is gelatin. Gelatin is just collagen. It's a protein. You have it melted, and you put the bacteria in it, and then you take a drop of this and put on some slide or something wherever you want to build your communities. You let this dry, and then you go in with a microscope and you isolate, find your favorite bacterium that you want to draw a house around, and then you draw the house. Now, remember, this is solid, so then what you do is you increase the temperature to 37 degrees. When you do this, everything you didn't cross-link melts, and now what you're left with, right, is a trapped microbe. The beautiful thing about this system is now it can be iterated. So now what you can do is you could take the same organism or a different bacterium, put this drop on top of this one now, and begin to structurally construct communities. And so this was actually, so using this technology, we've been able to actually do a lot of experiments. We don't really know anything about aggregates. We don't know much about their physiology. Are they like mushroom biofilms? Are they like planktonic cells? So you can do very simple experiments like this. This was actually done by Amy Wessel, whose picture is not up here for some reason, but she's right there. And she's actually works in Jean-Marc Guigot's lab now, and she was a grad student with me. And Amy was just asking the question, how big does an aggregate need to be until the oxygen levels drop? And it was a really important question, and the reason it was because in the Pseudomonas world, when Pseudomonas sees very low oxygen, it becomes resistant to antibiotics. And people used to attribute like the survival of Pseudomonas in wounds and in cystic fibrosis with giant aggregates that had low oxygen. But no one had ever asked the question, well, how big do the aggregates need to be till oxygen levels drop? And so Amy made a reporter that would turn on at about less than 2% oxygen. This is a glass slide. She filled up a trap of 9,000 cells. And what you can actually see is you don't see any GFP. This is full of cells. Although when she goes to 53,000 cells, then you can start seeing GFP come on, which means there's low oxygen. Of course, as an advisor, you're like, well, there are no glass slides in the lung or in wounds. So this is kind of irrelevant, right? Because you're on a glass slide. And so Amy actually put the trap up on stilts. So now it has a different diffusive surface. When you do that, you eliminate this, and now it takes almost 100,000 cells. And by doing this, along with modeling, she was actually able to begin to assess, what about these aggregate sizes, and are oxygen levels dropping? The other thing that we've been doing with the traps, I think, which is quite interesting, was begin to ask the question, so how close do 2 bacteria or 2 aggregates need to be to know the other's there? What's the limits of this? And of course, we can do modeling to actually look at this, but we need an experimental framework to do this. And so you can take responding populations and producing populations, change distance, change size, and ask when one can detect the other. And we've actually done quite a bit of this now. And this one is actually pretty cool because this is actually these experiments done in cystic fibrosis sputum. So one can actually use a natural environment to ask the question, you have a trapped population of Pseudomonas, and there are aggregates around it. Who can it signal to? And basically what we're finding is aggregates have to be extremely close together to communicate. And I think this is actually really important because there's a lot of ideas out there that people talk about long distance signaling and like you're signaling hundreds of microns away. And I'm not sure those data are actually, they certainly don't jive with what we're doing right now. And we see that we needed this really close proximity. And again, what it does is provide you with this experimental framework that now we can sort of move on. So the last slides are what we're doing now. And I've been telling, John Mark's probably tired of seeing this. I've been saying this for a year and a half, but these experiments are a little bit harder than I thought. That's a good thing about being a PI. We're basically looking at the spatial organization of populations in vivo. We're constructing them in the lab, putting them back into mice, and begin to ask the question, is spatial structure important? And do organisms know that they're misspaced? And if they are misspaced, what are the mechanisms by which you respace? Or do they do that? And these are sort of the things. The last thing I'll show you is, of course, there's one problem with this. And the problem with this primarily is that we have walls, right? We're going to put these traps in with walls. And what Jody did before she left the lab, she actually began to just construct traps that would dissolve with very, very specific half-lives. And so that's what we're sort of doing now. In the mice, so that you can structure populations, put them in the mice, allow them to dissolve, and then ask those questions. So that's my lab. That's what they think they look like. And this is Kendra Rumbaugh, who sort of works with us with Animal Work. So thanks.